Now in this section we will look at some of the carrier proteins and how they help other molecules to cross the membrane. Now, the examples that we saw previously, in which we saw the simple diffusion and osmosis, there was no energy involved. There was no ATP involved, and one of them was a simple movement of solute, and the other one was the movement of water across a semi-permeable membrane. However, items that cannot move freely in from the plasma membrane will move the membrane through these ways. And for the first example is called as, or the first mechanism or process is called as facilitated transport. Facilitated transport, as the name tells you, it's a transport in which you need a molecule which is facilitating the movement of one molecule to the other side. The facilitated transporters are always generally proteins, and they always move the molecules down a concentration gradient. Sometimes certain molecules that are just a little larger, and they may just need a facilitated transport. For example, glucose or amino acids will do it this way. So glucose and amino acids will not cross the membrane without a little bit of help, and the help is carried out by these carrier proteins. You can see this almost looks like a path or a passage that is shown over here, and the solute particles can get trapped, and then you can see how it pushes, the carrier protein pushes and makes ways for the glucose or the amino acid to move across. So this is with a little bit of help through these membrane proteins that will cause the movement of these molecules. Again, it is down the concentration gradient, that is important, and there is no ATP or no energy needed for this process, and this is called as facilitated transport. However, there are other molecules or ions that will move through the plasma membrane. Sometimes they're accumulating inside or outside, and they need to go the other way. For example, iodine collects in the cells of the thyroid gland, and glucose is completely absorbed from the gut by the cells of the digestive tract. And sometimes sodium can be almost completely withdrawn from the urine by cells of the kidney tubules. In all of these cases, molecules have moved to the region of higher concentration. Actually, it's going towards a concentration gradient. It's actually the opposite of what diffusion was. And because it's almost like taking a rock or a boulder and trying to move it up the steep hill. Now, if you move anything against a concentration gradient, then you always require energy, and the energy that is used over here is the ATP molecule. And ATP is the energy that would be needed to push the molecule against the force of gravity. And just like you have the water pump that pushes the molecules against the force of gravity, proteins will use the energy to remove a substance against its concentration gradient. Now, a very common example of this active transport is the sodium-potassium pump. Very important carrier protein are required, and in this one you can see on the first image that is shown over here that the carrier has a shape that allows it to take up the sodium. So notice when the sodium attaches, there are how many sodiums? There are 3 sodiums that are attached, and they are attached with the help of energy that is needed for sodium to attach to the carrier protein. Now, this is almost like a door that opens on the other side. So when the configuration of the carrier protein changes, it pushes the sodium out, and when it pushes the sodium out, the configuration now helps to take potassium ion in. So now it's going to take potassium this way, and it has thrown in sodium outside. And this way it is able to take this potassium and close again. Notice how it's going to take the potassium. So the potassium ion is going in, and 3 sodium are going out. And notice again it closes over here, and the phosphate group is again released when it closes, and the potassium ions are released to the inside of the cell. So this channel that is just created that allows the passage of ions against an electrical gradient or concentration gradient is due to a process which is called as active transport. Now, besides these, there are other molecules that can also cross the plasma membrane freely, and let's look. That's a third way of dealing with substances in and out, and this is due to the formation of what is called as vesicles. Vesicles are pretty much membranes that are in they invaginate, and sometimes they are just folds of the regular plasma membrane that will cause a formation of a vesicle or a bag, and these bags can either enter a cell or through a process which is called as endocytosis. Endo means inside. So notice that these plasma membrane is folding inside, invaginating, and it eventually closes it and throws the molecule in. This would be endo, it's taking in. And exo is outside. So sometimes molecules that are made, it could be even proteins molecules that are being made from the ribosome and that need to be transported somewhere away from the cell. And these will go out through exocytosis. Exo is outside. Even sometimes molecules such as waste that is generated by the cell needs to be released out. So in exocytosis, the plasma membrane, the vesicle actually is released, or it adheres to the cell surface, and it becomes incorporated in its intracellular matrix and then eventually thrown out. Now, endocytosis and can be either phagocytosis. Phago is also called as cell eating. So when solid objects are taken in, they're called as phagocytosis. Pinocytosis is when liquid goes in, also called as cell drinking, because the water is going in. Both of these processes definitely require energy. Endocytosis can also be receptor mediated, which means that there could be certain molecules of a receptor protein, as shown over here, that will mediate and help the formation of these vesicles. Once the vesicle is formed, it can be uncoated, and then it will eventually be released as lysosomes if it's an endocytosis. Receptor-mediated endocytosis is very selective, and it is much more efficient than just pinocytosis. It is involved in uptake and also in the transfer and exchange of substances between cells. For example, exchanges for example between maternal blood and fetal blood at the placenta is an example of a receptor mediated endocytosis.